Hello, everybody. Let's talk about sex a little bit. Eukaryotes produce either sperm or egg. Some plants and fungi can produce them both. But let's take humans as an example. Humans produce either sperm or egg. The germ cell line, or the diploid precursors to the sperm and the egg, are produced by mitosis. The gametes themselves are produced by meiosis. Meiosis is a final division. There is no cellular reproduction or of a single haploid sperm or egg once they're created. Once the sperm and the egg fuse, those two haploid cells create one diploid cell and meiosis can begin again. In this lecture, we will walk through the steps of meiosis and see how two genetically different individuals can produce offspring that resemble them both. Here we begin with interphase. Here we are. We know that the cell has just finished undergoing mitosis, so there is a duplicate set of DNA or diploid chromosomes in the nucleus. This has all occurred during interphase, and if this were a somatic cell, it would just continue the G1, S, G2 mitosis cycle to produce infinite number of daughter cells. But here in Module 6, we're talking about gametes. So after one round of mitosis, this cell is going to enter meiosis 1, beginning with prophase 1. Early in prophase 1, the homologous chromosomes are attached at their tips to the nuclear envelope by proteins. And recall that in mitosis, homologous chromosomes do not pair together, right? They line up randomly, but they're not paired. The tight pairing of the homologous chromosomes in meiosis is called synapsis, and we can see that happening here. We see the homologous non-sister chromosomes lining up next to each other. This is called synapsis. In synapsis, the genes on each chromatid of the homologous chromosomes are aligned precisely with each other. This supports the exchange of chromosomal segments between homologous non-sister chromatids, a process called crossing over. Crossing over can be observed visually after the exchange as chiasmata, which we can see here, and I forgot to include this term on the previous vocabulary list, so add chiasmata to your vocabulary page. So chiasmata is the point of exchange of genetic material between homologous non-sister chromatids. Let's take a closer look at what's happening in prophase 1. Here we see one set of homologous non-sister chromatids. So let's just, for the rest of this video, blue are the chromosomes from dad, red are the chromosomes from mom, okay? So we can see that these two homologous non-sister chromatids, so that's this one, which is, let's say chromatid A from mom is lined up with chromatid A from dad. So they're non-sister, but they are homologous. So we can see that these chromosomes are sort of sandwiched together, precisely aligned from tip to tip. And this is what we call forming a synapse when they're smacked together like that. Do you remember the name of the proteins that keep the sister chromatids attached here at the centromere? Do you remember? Did you say cohesins? Cohesins, right? So cohesins keep the sister chromatids together, and they also help with the attachment between the non-sister chromatids. Now that the homologous non-sister chromatids are locked into place, the first genetic variation event can occur, which is called crossing over, and that happens. We see the paternal chromosome and the maternal chromosome both containing genes for ABC. ABC, these are identical homologous sister chromatids. Little ABC, little ABC, these two are identical homologous sister chromatids. These two together are homologous non-sister chromatids with a gene for A gene for B, and a gene for C. During crossover, so these are aligned in a synapse, but we have them side by side for illustrative purposes here. So during crossing over, literal pieces of each chromosome are exchanged where they touch. So this is touching right here, which means that the resulting, as this process moves into metaphase, this piece of DNA is exchanged between each chromosome. So now the paternal chromosome has a little bit of maternal chromosome genetic material, and the maternal chromosome has a little bit of paternal genetic chromosome material. So after prophase, the two chromosomes are not the same as they were when they started prophase, and we see the first occurrence of genetic variation. A single crossover event between homologous non-sister chromatids leads to a reciprocal exchange of equivalent DNA between a maternal chromosome and a paternal chromosome. In this way, each gamete will carry some DNA from the parent and some DNA from the other parent. Each crossover event in each cell on each pair of homologous non-sister chromatids is unique. So the recombinant chromatid has a combination of maternal and paternal genes that did not exist before the crossover and is unique to that particular gamete. Crossover events can occur almost anywhere along the length of the synapse chromosomes. Different cells undergoing meiosis will therefore produce different recombination chromatids with varying combinations of maternal and paternal genes. Multiple crossovers in an arm of a chromosome have the same effect, exchanging segments of DNA to produce genetically recombined chromosomes. Okay, back to our schematic. We've undergone genetic exchange in prophase 1, so now we're ready to move into metaphase 1. During metaphase 1, the homologous chromosomes are randomly arranged at the metaphase plate, roughly in the middle of the cell. We've also called it the equator, with the kinetochores facing opposite poles. So the kinetochores on this red and this blue are facing this pole, and the kinetochores on this blue and this red are facing this pole. This random arrangement brings us to our second genetic variation event. Let's look at this more closely. Okay, here's an example of what I'm talking about. So for example, if the two homologous members of, let's look at this second chromosome set here. And let's call this, so they can line up in blue and then red formation, or red and then blue formation. And the same could happen to these. This is important in determining the genes carried by a gamete, as each will only receive one of the two homologous chromosomes when they split. So they're only going to, the ones on the left are going to go to the left, the ones on the right are going to go to the right. So remember that after crossing over, no two chromosomes are genetically identical anymore. And so this event here, where they can line up in blue-red or red-blue formation, is called independent assortment. Sometimes you may see it as random assortment. There are two possibilities for orientation at the metaphase plate. The possible number of alignments, therefore, equals 2 to the n in a diploid cell, where n is the number of chromosomes per haploid set. So human cells have 23 chromosome pairs, which results in over 8 million, so 2 to the n would be 23, which would result in over 8 million possible genetically distinct gametes just from the random alignment of chromosomes along the metaphase plate that we see here. The number does not include, so this variability in just this part in metaphase does not include the variability that was already produced by crossing over in prophase 1. So given these two mechanisms together, it is highly unlikely that any two haploid cells resulting from meiosis will have the same genetic composition. This is why some siblings look alike but don't look identical. In anaphase 1, we're here now. Here we are. In anaphase 1, the microtubules pull the linked chromosomes apart. So in metaphase 1, they lined up at the metaphase plate. In anaphase 1, the ones on this side go to this pole, and the ones on this side go to this pole. The sister chromatids remain tightly bound together at the centromere. In mitosis, they separated at the centromere. Remember, the cohesin broke down? But in meiosis 1, the centromeres remain intact and they're still attached to the microtubule spindle, and the whole thing, the whole chromosome is pulled over to one of the poles. The chiasmata that were formed in prophase 1 are broken here. So you see these attachments between non-sister homologous chromatids are broken when the chromosomes are pulled apart. Yeah, I think that's all I want to say about that. Okay. Now we round out meiosis 1 with a move through telophase 1 and cytokinesis, which occurs just as it does in mitosis. The nuclear envelope starts to reassemble, and the cleavage furrow is pinching together. One initial parent cell now becomes 2 daughter cells. One key difference between telophase in mitosis and telophase 1 in meiosis is that in telophase 1, each half or soon-to-be daughter cell contains half the number of chromosomes that the cell started with, but like mitosis, it's still a diploid set. So we see the 2 sister chromatids here. So each chromosome, these chromosomes are diploid, even though we started with 4 and now each cell only has 2. Now meiosis 2 looks exactly like mitosis. In prophase 2, the nuclear envelope that just formed here at the creation of these 2 daughter cells at the end of meiosis 1, in prophase 2, the nuclear envelope breaks down, and the chromosomes, if they had a chance to decondense, condense again. In metaphase 2, the chromosomes line up along the metaphase plate or the middle of the cell. And then in anaphase 1, the sister chromatids are pulled apart. The cohesin breaks down the centromeres, and the sister chromatids can be pulled apart to each pole of the cell, resulting in 2 new daughter cells after telophase 2 and cytokinesis. Now each daughter cell contains 1 chromatid from each set, or 1 set of genetic information, which means the final daughter cells in meiosis are haploid. Did you pay attention to see that in meiosis 1 we began with 1 cell and ended with 2? In meiosis 2 we began with 2 cells and end with 4. Some high notes to keep in mind when thinking about meiosis and meiosis 1 and meiosis 2 are that in meiosis 1, homologous pairs of chromosomes are held together at the chiasmata. So those are the touch points. These chromosome pictures are so cute because they all look like they're dancing to me. But remember, these touch points are chiasmata, okay? So the initial connection happens at the kinetochores and the centromeres, which sticks them together, but then there's physical connection and exchange of information at the chiasmata. And then in anaphase 1, homologous pairs of chromosomes are pulled by the kinetochore, so the centromere remains attached. The whole chromosomes are pulled apart, sister chromatids remaining attached to each other at the centromere. Then in meiosis 2, sister chromatids still held together by the centromere, but during anaphase, cohesin proteins start to break down, so the sister chromatids are then pulled apart, centromeres letting go, kinetochores moving, pulling each one to each side.